Hi everyone. Today we are examining a recent paper by Chama et al. That uncovers a novel function for the well-characterized nuclear protein MeCP2. This work positions MeCP2 as a direct inhibitor of the cGAS-STING pathway, a primary defense mechanism against cytosol DNA. We will explore how this discovery provides a new framework for understanding certain neuroinflammatory disorders.
We're going to follow the investigative path of the researchers. We'll start with their initial observation of MeCP2's interaction with cytosolic DNA, then move into the molecular mechanisms they uncovered, and finally discuss the physiological implications, particularly for Rett syndrome.
So, let's begin with the core observation that started this whole line of inquiry, the interaction of a protein that's supposed to be exclusively nuclear, MeCP2, with DNA found in the cell's cytosol.
Now, to appreciate the significance here, let's just briefly review the canonical function of MeCP2. It is a key transcriptional regulator located in the nucleus. Its main job is to bind methylated DNA and, in doing so, repress gene expression. And of course, mutations in the gene for this protein are the cause of the neurodevelopmental disorder Rett syndrome.
This brings us to the central question the researchers were asking. Could this nuclear protein, MeCP2, actually have a functional role out in the cytosol by binding to the very double-stranded DNA that's known to activate the cGAS-STING innate immune pathway?
To test this, they started with in vitro DNA pull-down experiments. The results, as you can see summarized here, were quite informative. MeCP2 showed a strong preferential binding to double-stranded DNA, and this affinity was comparable to that of the known DNA sensor, cGAS. This specificity is important, as it points towards a functional interaction, rather than just non-specific binding.
Of course, the next step was to see if this interaction actually occurs inside a living cell. So, they introduced tagged double-stranded DNA into cells and were able to co-precipitate both MeCP2 and cGAS. This confirmed that the interaction is not just an artifact of the in vitro system, but that it happens in situ. And this finding held up across multiple cell types, including both murine and human cell lines.
Direct visualization through immunofluorescence microscopy provided the next piece of evidence. After challenging cells with double-stranded DNA, MeCP2 was observed to accumulate in the cytosol, where it clearly co-localized with the DNA. This gives us direct visual confirmation of where this interaction is taking place.
So, we've established that MeCP2 is interacting with DNA in the cytosol. This naturally raises the question, how does a predominantly nuclear protein get into this compartment in the first place?
By tracking the location of MeCP2 over time following dsDNA stimulation, a clear pattern emerged. A few hours after the stimulus, MeCP2 began to appear in the cytosol. As the timeline shows, by the 6-hour mark, this translocation was robust, and you can see a corresponding drop in the nuclear signal. This change in localization was also sustained for at least 16 hours.
To determine if this was an active, regulated process, they used a chemical inhibitor of nuclear export called leptomycin B. The result was quite definitive. The inhibitor blocked MeCP2's accumulation in the cytosol, causing it to be retained within the nucleus. This provides strong evidence that MeCP2's exit is an active, regulated transport process.
A critical control was then performed. Is MeCP2's export a direct response to the DNA itself, or is it a secondary effect of the inflammation that the DNA triggers? To test this, they used cells that lack cGAS, and even without the primary DNA sensor and the subsequent signaling, MeCP2 still moved to the cytosol. This demonstrates that the cytosolic dsDNA is the direct trigger for the export.
And importantly, this mechanism isn't limited to externally introduced DNA. The same phenomenon was seen in TREX1 deficient cells. These cells are known to have a chronic buildup of their own endogenous DNA in the cytosol. This is a key point, as it implies MeCP2 has a role in regulating immune responses to self-DNA, which has clear implications for autoimmunity.
So we've established that MeCP2 moves into the cytosol and binds DNA. The next logical step is to ask, what is the functional consequence of this? Given that both MeCP2 and cGAS are in the same place and bind the same substrate, the hypothesis of competition naturally arises. Are these two proteins competing for access to cytosolic dsDNA?
The evidence for direct competition is compelling. In cells that lack cGAS, the amount of MeCP2 that could be pulled down with dsDNA went up. And conversely, When MeCP2 levels were knocked down, more cGAS was able to bind to the DNA. This reciprocal relationship, which is illustrated here, confirms that they are in direct competition for the same molecular substrate.
This competition has a significant functional outcome for pathway activation. In cells that are deficient for MeCP2, the cGAS pathway becomes hyperactive. It produces over 4 times the amount of its enzymatic product, 2,3-cGAMP. This indicates that MeCP2's normal function is to dampen the catalytic activity of cGAS.
This elevated cGAS activity translates into a hyperinflammatory cellular state. We see increased expression of type 1 interferon genes and other inflammatory markers. The whole inflammatory signature is dependent on this pathway, as it can be eliminated with a STING inhibitor. And notably, this effect is specific to the DNA sensing pathway. Immune responses that were triggered by RNA agonist were not affected by the absence of MeCP2.
So, having established the molecular regulation, let's now consider the physiological relevance. Does this mechanism actually change how a cell responds to a real viral infection?
The data from viral infection models is consistent with this hypothesis. During an infection with the DNA virus, HSV1, MeCP2 was exported to the cytosol, but this didn't happen during infection with an RNA virus. As a result, cells that lacked MeCP2, and therefore lacked this inhibitory break, were actually more resistant to the DNA virus. This demonstrates a clear physiological effect on the antiviral state.
This phenotype is also supported by transcriptomic data. Gene expression analysis from cells, from mouse models, and from human patients with Rett syndrome all show a common signature: a significant upregulation of the type I interferon response pathway. This suggests a kind of basal heightened state of antiviral alert when functional MeCP2 is absent. But the direct inhibition in the cytosol is only one part of the story. The research also uncovered a secondary, indirect mechanism that helps to sustain inflammation. It's a feedback loop that originates from MeCP2's primary function back in the nucleus.
To understand this loop, we just need to recall MeCP2's canonical role. One of its key functions in the nucleus is to repress the transcription of endogenous retroelements, like line 1s, which are essentially remnants of ancient viruses that have been integrated into our genome. This leads to the next question: what happens to these line 1 elements when MeCP2 is no longer in the nucleus to keep them in check? Does its departure from the nucleus allow them to become active?
And the data confirms this hypothesis. When MeCP2 is exported from the nucleus after a dsDNA challenge, the transcription of line 1 elements goes up significantly. This de-repression appears to be a direct consequence of MeCP2 being relocated from its usual site of action.
And this is where the feedback loop really ignites. The newly transcribed line 1 RNA is then reverse transcribed back into DNA by its own machinery. This process generates a substantial amount of new endogenous dsDNA right there in the cytosol, which then serves as fresh fuel for the cGAS-STING pathway.
So let's just summarize this self-perpetuating cycle. An initial dsDNA trigger causes MeCP2 to leave the nucleus. This leads to the de-repression of line 1 elements. The activity of these elements creates more cytosolic dsDNA. And this new DNA then further stimulates the cGAS-STING pathway, creating a positive feedback loop that sustains the inflammation.
When you put all of these findings together, they converge on a new model for Rett syndrome, one that provides a mechanistic explanation for the chronic inflammation that's observed in this disorder.
We can now think about a dual role for MeCP2 in immunity. In the cytosol, it acts as a direct brake on the initial immune response, and in the nucleus, it acts as a gatekeeper, preventing that inflammatory feedback loop from ever getting started. In Rett syndrome, where MeCP2 isn't functional, both of these control mechanisms are lost, and the result is chronic immune activation.
This model also suggests a potential therapeutic intervention. The researchers show that by inhibiting line 1 activity with reverse transcriptase inhibitors, they could interrupt this inflammatory cycle. This highlights a potential new therapeutic strategy for mitigating the inflammatory pathology in Rett syndrome.
This work, therefore, has significant implications. By establishing a direct mechanistic link between MeCP2 and dsDNA-driven inflammation, it not only reframes our understanding of Rett syndrome, but also identifies potential therapeutic targets for a broader range of neuroinflammatory disorders. The future of treating these conditions may well involve modulating this newly defined axis of innate immunity.